The video is for selection of the chromatographic conditions, chromatographic parameters, selection of the mobile phase, column, and the detector for the HPLC analysis. I welcome all the viewers to this video on how to select the right mobile phase, column, chromatographic conditions, parameters, and detectors in the HPLC. HPLC is the widely used analytical technique in the pharmaceutical industry, and proper selection of these parameters, columns, mobile phase detectors is very important because it directly affects the method's accuracy, precision, robustness, specificity, and all the validation points as well. First is mobile phase selection. So as we know, HPLC is a chromatographic technique in which the stationary phase is there and mobile phase is there. The stationary phase is present in the column. The analyte is supplied to the stationary phase through mobile phase. In the column, the separation happens and detector records the response. This is the very basic idea about the HPLC technique. So, selection of the right mobile phase involves the understanding about the chemistry of the analyte and principles of the HPLC. The mobile phase must ensure good solubility, stability, and peak shape. It should be compatible with the column chemistry and must be UV transparent if you are using UV detector. There are certain types of there are some types of mobile phases like organic solvents you can use, acetonitrile, methanol, aqueous phase buffers you can use, water you can use. Acetonitrile is with low viscosity and strong elution strength it is having. Methanol is cost effective and has the higher viscosity. Aqueous phase buffers in the pH range of 2 to 7 and water HPLC grade can be used. Then how to select the mobile phase? So mobile phase is selected considering the solubility, considering the pK of the drug, log P of the analytes. pH should be in the variation limit of 1 unit from the analyte pK. We need to avoid the mobile phases with pH greater than 8 in the silica base C18 columns. Use the buffer strength proper with the requirement. Check UV cutoff for the solvents. Column selection. Second is the selection of the stationary phase. So we should know the column chemistry, chemistry of our analyte, and how the separation will happen. Stationary phase chemistry involve the columns with a C18, C8 phenyl group, cyano group, or simply you can say C18, C8 phenyl and cyano. Particle size of 3 to 5 micron. Pore size is important as per the small molecules and the large molecules like peptides and proteins. Column dimensions, so medium length or longer length column compared to the short columns. So column dimensions, column chemistry, and the particle size impacts. So the selection criteria is based on that. C18 is mostly used for non-polar compounds. C8 is for slightly polar compounds with shorter retention. Phenylhexyl for aromatic compounds with pi-pi interactions. Cyano for polar analytes, normal and reverse phase. Amino columns for sugars, carbohydrates. Then chromatographic parameters. So it involves the injection volume, temperature, flow rate. Flow rate typical 0.8 to 1.2 ml per minute. Higher flow will give you faster run but will have the lower resolution. Column temperature 25 to 40. It improves the reproducibility and peak shape. Injection volume 5 to 20 microliter. So depending on to your chromatographic conditions, you can select that. You can go for gradient versus isocratic. Isocratic means single mobile phase composition for a simple analyte mixtures, and gradient for those which are complex formulations or complex analytes. In gradient, the composition of the mobile phase changes over time, needed for the complex formulations, reduces runtime and improves the resolution. System suitability parameters to monitor: resolution, tailing factor, theoretical plates, percent RSD of 5 injections, retention time, stability. These are the system suitability parameters. In upcoming time, I will be making the separate video on system suitability to let you know more regarding that topic. Then detector. So based on your analyte, its chemistry, the detector is selected. UV visible detector is most common and it is used and it works for the analytes with chromophores. We need to select the lambda max after scanning the analyte. PDA photodiode array, peak purity, identification of co-eluting peaks, multivalent detection is possible with PDA. Other detectors like refractive index detector, fluorescence detector, ELSD, CAD, these are the detectors used. Fluorescence detector is highly sensitive and requires derivatized fluorescence. Refractive index detector for the compounds without UV absorption cannot be used in the gradient way. So if you select all these parameters, the mobile phase, stationary phase and the chromatographic parameters, then you can develop a suitable method and validate it. Mobile phase selection is based on the solubility of the analyte, pKa and the UV cutoff. Column is based on the chemistry of column, chemistry of the analyte, particle size and dimensions. Parameters like flow, temperature, gradient and system suitability parameters. Detector depend upon the analyte's detectability. So this is the information regarding the HPLC and selection of the right mobile phase, stationary phase and the parameters. Thank you for watching the video.